Hello, everyone. I'm Ari Kozik, a research investigator at the University of Michigan in the Division of Pulmonary and Critical Care Medicine. My current research focus is the role of the respiratory microbiome in adult asthma phenotype and treatment response. Other interests include translational systems biology and biopsychosocial models of disease. I'm also the co-founder and vice president of the Black Microbiologists Association. Today, I'm going to briefly discuss the human microbiome in the context of health disparities and the critical need for a conceptual shift in the way we think about this issue. First and foremost, microorganisms are everywhere. They inhabit nearly every ecosystem on the Earth. Much like us, they often exist in communities. A collection of microorganisms and their genes that inhabit a particular environment is called a microbiome. There's ongoing study of the microbiome of almost every ecosystem you can think of, whether that be Arctic ice, animals, plants, and of course, humans. With the help of technology, we are gaining a profound appreciation for the global ecological significance of microorganisms. If you're like me, you grew up thinking about microbes as germs that were bad for you and caused disease. To be fair, many of them are quite unpleasant and lead to things like the flu or COVID-19. But we also know that there are non-pathogenic microorganisms normally associated with nearly every body surface. These microbes help us digest our food, adjust to our environment, and train our immune systems. When people talk about the microbiome, they usually mean bacteria, but your microbiome is more than just bacteria. There are also viruses, fungi, archaea, and in some cases, parasites that are associated with our bodies. Your microbiome is unique to you and responds to your environment and exposures throughout your lifetime. We're only just beginning to understand the rules that govern these functions, but thus far, the data is leading us to think differently about health and disease. One of the big biomedical challenges we face today is figuring out how the microbiome is involved in human health and disease. The human microbiome has been implicated in several diseases and disorders, many of which impact certain populations disproportionately. Microbiome research is a rapidly developing field, but it has become clear that in order to fully understand the impact of the microbiome and further its potential for the restoration of health, we must recognize it as an ecosystem that is complex, dynamic, and responsive to its environment. Technological advances now allow assessment of interactions within communities of microbes and between microbes and their hosts. So can we learn from the microbiome? Can it help us understand the complexity of disease processes across individuals and populations? Can we modify it to restore health? I believe that we can, but current evidence suggests that we must take a step back to consider the human microbiome for what it is, an ecosystem in constant communication with an environment that itself is continually shaped by larger biological, social, and environmental forces. It is no secret that vast disparities in disease burden exist across the human population, especially among the poor and individuals from marginalized racial and ethnic backgrounds. This is a critical problem in the United States and globally. There have been renewed calls to work towards health equity, which will require an integrated, multidisciplinary approach. Historically, racial and ethnic classifications have been socially constructed based on perceived biological differences and used as justification for systems of social hierarchy and oppression. Race-based biological or purely genetic hypotheses and investigations have failed to explain or eliminate health disparities. Conflation of race with biology and the study of health disparities promotes research priorities that focus on identifying individual or group deficiencies while minimizing or ignoring larger systemic environmental and structural forces that can influence outcomes. This framing is common in the literature. In this study, microbes and their functional capacities from multiple body sites were compared across select racial categories. The authors report racial and ethnic background proved to be one of the strongest associations of pathways and microbes with clinical data. They also mention an association between race and vaginal pH. One of the many problems with this kind of framing is that there isn't sufficient follow-up discussion to interpret what those associations may mean, and we're left with an important question. Are these microbial differences inherent in racial categories? I would argue that the implied answer from the omission of further discussion is yes. From this kind of implied reasoning, studies of microbial communities across populations, especially those pertaining to the human microbiome and health disparities, uphold a flawed model where race determines some sort of biological state that then leads to poor outcomes rather than understanding race as a convenient measurement that should direct us to closer examinations of social, environmental, and structural contexts. Currently, we've made some progress in conceptualizing environmental impacts to our microbiomes. Until recently, we were thinking and writing about this at a very granular level, focused on individual and group social behaviors and environments. While we've begun to incorporate other domains like the natural ecosystem and the built environment, our consideration of the impact of race and racism has typically been limited to behaviors, body sites, or group-level differences in microbial community membership. However, humans are an ecosystem within other ecosystems, and racism by design alters our experiences of and access to these systems. So in order to restore dysbiosis, we must shift our perspectives to understand its cause and examine the impact of racism in all domains. For example, I'd like everyone to think back to the sensory memories that define their childhood. Was it the smell of fresh cut grass? Perhaps the smell of soil? Maybe you live near a lake or someplace with fragrant trees, or maybe the distinct smell of smoke from bonfires or fireplaces or leaf piles. For me, the most vivid are the industrial odors that defined my memories of my early grade school years in Northwest Indiana. I remember playing at the school ground in this community of color each morning. The air was thick and acrid, the smoke visible even on sunny days. The mist of sharp industrial odors from the nearby refineries, mills, and smelters filled my nose and lungs, impossible to ignore, yet mingled with the smells of breakfast from the cafeteria. Our family was fortunate enough to be able to move away from the industrial shoreline, but every morning arrival at school was marked by this experience. After about half an hour or so, a headache usually followed, along with a slight but persistent nausea that would keep me company all day. Many of my schoolmates had asthma. They would go to the nurse's office at regular intervals for their inhalers. It seemed like the norm. Those smells had a source. This community along the shores of one of the Great Lakes is home to around 200,000 people, but also some of the nation's largest integrated steel mills, refineries, and smelters. Heavy industry blossomed in this area after World War II, tightly linking people's economic stability to the expansion of industry. As pollution increased and the health risks that come along with it, population shifts accelerated during the 50s and 60s, with wealthier middle-class white individuals leaving for more suburban, less polluted areas. Today, due in part to the history of discriminatory housing practices, these areas are predominantly communities of color. The health consequences of this environmental injustice are dire. I mentioned that asthma was common among my peers. For my mother, an educator who spent her entire career in this area, cancer was common among her peers. These anecdotes are reflected in the data. A 2009 study found that air quality outside of these schools exposed children to higher levels of airborne toxins, including a variety of metals, than anywhere else in the US. The water supply and recreational waterways are also similarly impacted by pollution. The state ranks ninth in the US per capita cancer deaths. These issues, long suspected among residents of the area, finally came to light in the last few years when the housing complex was evacuated after residents, including children, tested positive for toxic levels of lead exposure. The housing complex was built on the site of a former smelter and was finally determined to be uninhabitable 30 years after the site was known to be contaminated. Generations of predominantly Black and Hispanic residents in this area have suffered health issues and increased mortality as a result. These kinds of stories are not isolated. Similar trajectories have been uncovered across the US in places like Flint, Michigan, and elsewhere, with communities of color facing a disproportionate burden of many chronic diseases. Under the conventional framing, studies that would seek to decipher the role of the microbiome in, say, chronic lung disease disparities may sample from these residents and make any number of comparisons, but without considering their environmental exposures and other systemic and structural features that can impact their microbiomes, we miss a huge part of the picture. And this is increasingly demonstrated by the persistence of the disparities. So then what do we do? How do we understand microbial contributions to health disparities and restore dysbiosis? We must expand our models to reconsider the sources of dysbiosis. The data is clear. Racism and discrimination alter our experience of societal structures. Therefore, we need to understand how the microbiome interacts with these factors if we want to decipher its role and work with it to restore health. This frame shift will undoubtedly be complex and challenging, but it is critical. Studying and understanding these systems is critical to our work towards equity. As a field, microbiology has a lot of work to do. How do we address racism as a source of dysbiosis in studies of the microbiome and health outcomes? How do we collaborate across disciplines to do this work? How do we get funding for that work? And then, of course, how should our science inform and impact the systems and structures that made the work necessary? It's a monumental task, to be sure, but as scientists, we must not allow the complexity of these issues to preclude progress. Thank you for your attention and your attendance. I'm happy to discuss further at the live session.